Dear students, let us discuss another topic, that is DNA repair. The mechanism of DNA repair. We have discussed till now various mutations in the DNA, and there is a mechanism present within the living cell which repairs most of the damage of the DNA, and which is called the DNA repair mechanism. In order to understand DNA repair mechanism, you must understand the structure of the DNA, that the DNA is made up of the two strands. So, DNA is double stranded and the strands are present in a helical fashion. Each strand is having a phosphorus sugar backbone. So, if you look chemically, you will find that phosphorus is linked with this sugar molecule, then sugar with the phosphorus, phosphorus with the sugar, sugar with the phosphorus. So, this form the strand of the DNA. So, you have one strand, then the other strand. Both the strands are then joined by one another with the help of the pairing of the bases, that in between these two strands you have the, say for example, if adenine is there, the complementary pair of the adenine is the thymine, and if thymine is there, then it will be adenine. If cytosine is there, the complementary pair is guanine, and if guanine is there, then you have the cytosine. Guanine and cytosine, they are paired with one another with the help of the hydrogen bond, and accordingly 3 hydrogen bond, and the adenine and thymine are paired with double hydrogen bond. Now, the bond present between this phosphate group and the sugar, that means this one, this is called as phosphodiester bond, and the bond present between sugar, as you know, the bases are linked with the sugar molecule of the strand. This is called as glycosidic bond. So, you are having the two bond between phosphate group and the sugar molecule, phosphodiester bond, and the sugar molecule and the base, which is called as glycosidic bond. DNA repair is a collection of process by which a cell identifies and correct the damage to the DNA molecules. So, it is not a single process, you have so many processes which correct the DNA molecule after identifying the damage. You know, DNA is damaged by so many metabolical activities of the body as well as environmental factors, which may includes the physical factor like radiations, the chemical factor like chemicals, and on a day there may be thousands of damage to the DNA of the cells, but you will find that all these damages are not carried forward, as they have some repair mechanism present in the cells also. It is the repair mechanism of the DNA which is responding constantly to the damage of the DNA, to the damage to the DNA caused by these environmental factors and the metabolic activities. As a result, the total mutations within a cell is kept under tolerable level. It is not allowed to go beyond the level of the tolerance. So, the cells survive even after having thousands of mutations per day because of this repair mechanism. The rate of the DNA repair in a cell depends upon many factors like what is the type of the cell. There are some cells which may not have the good repair mechanism. So, once they are damaged, then the damage may not be recovered. Then the extracellular environment is also important, because If the extracellular environment is having plenty of mutagens, then the mutation rate may be higher than that of the this repair mechanism. So, what will happen that the cell will not be able to repair to a considerable level, and accordingly if the cell is old enough, its age is very high, it means again the cell will not get repaired. Now, if cell will not repair itself, what will happen that the mutations will get accumulated in the cell, and if the mutations are accumulated within the cells, so what will happen, cell will not be able to survive. So, either it has to go senescence, that it will undergo irreversible state of dormancy, so that it may not function normally, because normal function is not possible due to the heavy damage of the DNA, which has not been repaired because of the defective mechanism, or because of the type of the cell, because of the age of the cell, or because of the extracellular environment having high quantity of the mutagens, or cell may go to suicide, which is called apoptosis or programmed cell death, that the cell go die, so that it may not replicate the high level of mutagens in the newly formed cells. So, that because the mutation has reached to a level which is not tolerable to the cell now, and or the cell may go unregulated cell division. That means, if it will go under division after having such heavy mutations and not repairing mechanism, and having not repairing mechanism, then what will happen, that if such type of cell will divide, then it will not be a normal division, it will lead to the form of the cancerous cells. The DNA repairability of a cell is vital to the integrity of its genome, and thus to the normal functionality of the organism. So, if organism wants to have the normal function, then it means repair mechanism must be effective enough. As far as the mechanism of the DNA repair are concerned, repair mechanism of the DNA are universal. They are present in the microorganisms, and they are present in the even up to a higher animals like human, and almost similar type of mechanism present in all. Some common mechanism of DNA repair are: one is photoreactivation or the direct repair, other is the mechanism which are involved with the repair of the single strand of the DNA. When only one strand of the DNA is damaged, then the other strand can be used as a template, so to guide the correction, because DNA strands are complementary to each other. If on one strand it is adenine, then on the opposite strand it will be guanine. Say for example, if the second strand is damaged, then the original strand may be used as a template, and the guanine may again be reinserted to correct the damage of the DNA. So, it is the single strand repair, you can say. You have so many methods to repair the single strand, like excision repair, photorespiration repair, post replication repair, uracil DNA glycosylase repair, mismatch repair. But sometimes what happens that both the strand of the DNA are damaged, and this damage is often very hazardous, because there is no other strand present which can guide the repair of the damaged part. So, it will always lead to a genome rearrangement. So, that is why it is the hazardous double strand repair. Double strand damage is often hazardous. There are some mechanisms which are associated with the repair of the double strand of the DNA also, like the non-homologous end joining, NHEJ, Microhomology-mediated end joining, MMEJ, and homologous recombination HR. Let us look each of them one by one. Now, the photoreactivation or direct repair. It is called direct repair or the photoreactivation, because the enzyme that is involved in the repair like photolyase is requires this enzyme require energy in the form of the light, that to the blue light of the visible spectrum uh, to get its activity. So, it means this enzyme is otherwise present in inactive form, but after absorption of light it will get activated and it will perform the function. So, it is called photoreactivation, and it is called direct repair because no new synthesis of the part of the damaged DNA will takes place. It will just reverse uh, the change that has taken place. Let us take an example. For example, you are having this DNA molecule which because of the UV radiations has got the thymine dimer within this strand. Then what happens during the dark, this photolyase enzyme bind with this DNA, but because with this thymine dimer particularly it recognize the site of the thymine dimer, then it will bind to this site, and after absorption of the light this enzyme will get activated, and after activation it will break the bond between the thymine to thymine. Thus, it will reverse uh, the dimer formation. As a result, after breaking this bond, it will get itself released from this DNA molecule and the DNA molecule will become normal. So, because no new synthesis is there, so this is called the direct method also. Similar type of this enzyme also act upon the cytosine dimer, the cytosine thymine dimer also, and this photolyase is an enzyme which is normally present in bacteria, fungi and most animals, but in human being this type of the mechanism is not present. In the human being, the correction is taken place with the help of the nucleotide excision repair, which we shall discuss in the next slide. Now, nucleotide excision repair, as the name itself is indicating, you have to remove some nucleotide and then you have to repair, you have to resynthesize that part. Let us take an example. The first step is the incision step. What will happen that when the DNA is damaged by the UV radiations, say for example, thymine dimer is there, then the endonuclease are the enzymes which will recognize this thymine dimer, they will bind to this thymine dimer and then they will cleave, they will make a cut in the strand of the DNA. That means they will break the phosphodiester backbone. Now, you can recognize the phosphodiester bond, that means the bond between the sugar and the phosphate group of this DNA strand on both side of this thymine dimer. After having this break on both the side of this thymine dimer part, the exonuclease property of the enzyme will then take away this part, will remove this part, and then the DNA polymerase will resynthesize this part by using this part as a template. That means, if adenine is there, then thymine will be added opposite to it, opposite to this thymine adenine will be added, opposite to this cytosine guanine will be added, opposite to this thymine adenine will be added, likewise thymine and thymine opposite to these two adenine. And once this new strand is synthesized using this DNA polymerase, then the next step of the enzyme, that is in the next step DNA ligase will bind this part with the previously synthesized part of the DNA, and thus they repair this part. Now, it means this first step was the incision, that is making a cut, then the removal of this part which is called the excision step, and then there is a synthesis of this part which is called the synthesis step, and then there is a binding step taken over by the DNA ligase. Now, this excision nucleotide excision repair sometimes repair only a very small part of the DNA, that means they repair only one base or few base, then it is called the very short patch repair VSP, and if it repairs a moderate size of the DNA molecule, DNA part or the DNA segment, then it is called the short patch repair, and sometimes it repairs a very long part, then it is called the long patch repair. Then you are having the post replication recombination repair. Now, it involves repair of replicated but damaged DNA followed by the recombination. That means, DNA is already replicated, but the replicated part of the DNA has some certain damage in it. So, that is why it is post replication, that it will be repaired after replication, and then the mechanism which repair them is the recombination type of the repair. We shall look into it. Say for example, if this the DNA molecule, it is irradiated by the UV and diamine dimers are there, but you see both the strands are affected. This strand is affected over there, this strand is affected over there by the formation of the diamine dimer. What will happen that during the replication by the DNA polymerase, what will happen that opposite to this diamine dimer gap will be formed. So, again once this strand will be synthesized, this strand will synthesize the new strand. Say for example, it was having diamine dimer over there, then opposite to this the new strand will have the gap. So, again this strand was having diamine dimer over there. So, once this strand will replicate, the opposite strand will have the gap over there. So, in this way the newly replicated DNA molecules will have the dimer on one side and the gap on the other side, because after replicating up to certain point, when the dimer is detected, polymerase is unable to replicate this part. It will leave this part of the DNA and will join the part after crossing the dimer part and will synthesize the rest part of the DNA. So, it means you will having the DNA molecules having dimer on one side and the gap on the other side. What will happen that a very unusual type of recombination sometimes happen. What happens that both the affected strand come together and both the normal strand go to other end. That is, if the two sister chromosomes recombine such that, because you know this is one chromosome, this is other chromosome, they recombine in such a fashion that the dimers and the gaps end up in one chromosome. That means, one chromosome may become totally you can say non-viable or the damaged, having damaged DNA, and the recombination lead to the formation of the other chromosome, which will have all the both the strand of the DNA intact. Say for example, the recombination between these two chromosomes will takes place like that. Say for example, this chromosome was having this part intact and this chromosome was having this part intact. If this part will join this part, so it will become an intact strand at a total, and accordingly if this part will join this part, then the second strand will become totally intact. On the other hand, this portion having diamine dimer join the part having the gap also. So, we will having the thymine dimer and the gap, and accordingly this diamine dimer will join this gap part. So, it will have diamine dimer and the gap. So, one chromosome is non-viable, whereas other is wild type. So, this is an unusual type of recombination sometime that takes place after the replication in order to correct, because otherwise it is not possible to correct both the strand at a time. So, it chooses a way that let one of the chromosome be totally defective and the other chromosome be totally repaired and totally correct or the wild type. So, this is called the post replication recombination repair. Then you are having the uracil DNA glycosylase repair. You know, uracil is produced in the DNA if the cytosine is deaminated, that is amino group of the cytosine is removed, and you know that uracil is not the part of the DNA, uracil is the part of the RNA. Uracil, you have the enzyme called the uracil DNA glycosylase enzymes. It removes the uracil from DNA, and so the name of this enzyme is by cleaving the glycosidic bond. So, glycosidic bond is a bond present between the nitrogenous base and the sugar molecule of the strain. So, it removes this uracil by cleaving the glycosidic bond, and once the uracil is removed, it means the DNA will have the A pyrimidine site on one strand, and such A pyrimidine site have been shown to undergo an axion repair mechanism that we have already discussed. So, after removing the uracil, again similar thing will happen as we have discussed in the axion repair mechanism, that by using the other strand as a template, new segment of the DNA will be synthesized, which will be ligated with the help of the ligase enzyme, and the repair will be done. Then you are having the mismatch repair or the error correction during replication. Now, what happens? You know that during the replication, one strand acts as a template and the other strand is synthesized on that very strand by reading the frame, because by reading the frame, the DNA polymerase can substitute the corresponding base at the opposite end. So, new strand is synthesized during the DNA replication by reading the complementary base pairing. So, this new strand is also called as nascent strand. What happens sometimes that if the new strand, say for example, over here opposite to thymine, adenine was supposed to be added over there, but in the new strand due to some mistake guanine is added. Now, the question is, both the strand are having the base pair, they are not having any deleted base or inserted base, neither they have any break nor they have any dimer. Now, the question is how to recognize which of them is correct. So, there is a very usual phenomenon in the DNA, that is the methylation. The methylase enzyme does methylation of the adenine. So, before the replication at the region which is called as GATAC. GATAC means GATC, whenever it found GATC, it methylate this adenine. So, say for example, originally this was the template strand. So, adenine of this is always methylated. So, it means once the new strand is synthesized, until the methylation will not take place, this new strand will be recognizable, because the question was which of these strand is correct. So, obviously, the new strand is incorrect. No, in order to recognize the new strand, the DNA polymerase what does DNA polymerase recognize the old strand by having the methylation of the adenine at the GATAC, and recognize the new strand by not having the methylation at the GATAC and GATAC sequence on the adenine. So, once the new strand is recognized by the enzyme, then again similarly the endonuclease will come, they will just cleave the mispaired, mismatched base opposite to the old strand, they will cleave, then the exonuclease will remove this part, and then DNA polymerase will resynthesize it, and the ligase enzyme will seal it, and the original strand will be original or the repair of the strand will be done. So, here what I mean to say that there was a mismatch, and it was the methylation which was used to recognize the old strand and the new strand, and once the new strand was not having the methylation on the adenine at the GATC sequence, that means it was the new strand and it was corrected. So, this is the mismatch repair or the error correction during replication. Then you are having the non-homologous end joining. So, what happens? that if the if the break takes place between the two strands, so then the mechanism which is involved to anneal them or seal them is called the non-homologous end joining. It is called non-homologous end joining because the break ends are directly ligated with the help of DNA ligase without the need of a homologous template to guide repair. That means no homologous segment was present over there. So, it was just annealing of the two broken end. So, there was no need or no utilization of the this homologous template. So, this is just annealing of the two broken part. This is called the non-homologous end joining. This is relatively accurate, although small amount of the deletions or insertion may takes place after this re-annealing. So, it means not 100% correct, because I have already told you that the double strand break are often lead to some rearrangement in the DNA, because it is not 100% accurate mechanism is present, not accurate mechanism is present to this repair the break in the double strand DNA, because in the single strand DNA you are having an edge that if the break has taken place on this strand, then the you can say the opposite strand is intact. So, the complementary base pair may guide you that what is to be added over there, but in the double strand DNA you are missing this thing. As a result, you are always having some deletion or the insertion of the some nucleotide. Then so, this simple annealing sometime may lead to the translocation in the chromosomes and the telomere fusions also, which may lead to the tumor cells. Then you are having the homologous recombination. Homologous recombination means you are having double break in this chromosome, but you know every individual is having the two chromosome. So, after having replication in the S phase, after having the synthesis of the DNA in the S phase, you will have the two chromosome, that means you have the double amount of the DNA. So, if one chromosome, or say for example, this is the one chromosome, it is having its homolog pair, the one pair is having intact DNA and other is having double strand break, it will utilize the this homolog pair, the other chromosome of its pair to synthesize this missing part. So, by doing so, it is utilizing some sort of homologous base pairing on the opposite chromosome. So, it is called as homologous recombination. The machinery involved in this is exactly like that of the machinery which is involved in the crossing over of the chromosome, because this is exactly a mechanism. You see this part was deleted in both the chromatid of this chromosome. So, what is happening over there, that this part is coming down to have the bases, complementary bases of this intact chromosome, and then after getting this direction from this, it get repaired, the new strand is the new part of this broken DNA is resynthesized, and then it will go back and will join with the help of the ligase enzyme with this. And again this will again take the help of this as a template strand. So, the broken part will be resynthesized. This is called the homologous recombination. Then you are having this microhomology-mediated end joining. Now, basically whenever there is a break in the two strand of the DNA, it is not necessary that break will always be vertical. Say for example, break may be of such a kind that you are having this part having an overhang of the below strand, and here you are having the overhang of this strand. So, what happens in this mechanism, the two strands will be pulled together by the enzymes. When they will be pulled together, they will have some sort of microhomology. that once this strand will approach over there, it may find some bases which are exactly complementary to one another, and then it will pair with one another. This is called microhomology. Once the microhomology is there, it will pair with one another. Then the part which is present next to it, like this part of this segment, which was not finding its complementary base, because due to this break some deletion might have taken place, and it will be flanked. It will not be allowed to join with the other part. It will be flanked above with the help of the nuclease enzyme and will be removed. Then what will happen, that the DNA polymerase, after joining this microhomology part, will start synthesizing this part by using this segment of the DNA as a complementary strand, and again in this direction also it will synthesize this part by using this strand of the DNA as a template strand to have the bases added over there. So, as a result, after synthesis of this portion and this portion, by utilizing opposite strand, it will get ligated one another, and this flanked part will be removed and DNA will be repaired. This is called as microhomology mediated end joining. Again, because this part will be deleted, it means you will not have the 100% accurate repairing. So, some deletion might takes place. As a result, the chromosomal abnormality may takes place over there, DNA abnormality may takes place over there, which may lead to the certain abnormal conditions in the cells. These are the various mechanisms which were involved in the DNA repair. So, in order to understand the DNA repair, you must have the accurate knowledge of the DNA replication, because DNA replication is the method which is used by most of the methods in the DNA repair. So, that is how the complementary strand is beneficial, how the complementary pairing is done during the DNA replication. Likewise, you must have the role of the enzymes like the polymerase, like the ligations. All these concepts must be clear to you, so that you may understand all of these mechanisms very clearly. Thanks. This is all about the DNA repair mechanism.